Warm welcome to everyone. I am VEDHALAKSHMI RAJAN and I am currently doing BE Bioinformatics at Sarita School of Engineering. And I am here to present the manuscript on the topic of the tear proteome analysis in molecular surface diseases using label-free LCMS and multiplex microarray biomarker validation. The paper is written by Xavier Sorolla, who is the main author of this paper with his teammates. Xavier Sorolla is currently has written this paper from the Bioinformatics Applied Research by Baraskia Science and Technological Park in Baraskia, Spain. So the background of the literature. This focuses on investigating the protein composition of the tear of the patient with OSD, such as dry eye diseases, that is DED, and meibomian gland dysfunction, MGD, which is compared with the normal volunteers where we represent them with the CT. Here, in this literature, the potential of advanced proteomic techniques, which helps in understanding which helps in advancing our understanding and management of the ocular surface diseases. The integration of label-free LCMS and multiplex microarray technology provides a comprehensive approach to the understanding of the tear protein. The functional network analysis reveals the main biological process that should be examined to identify the biomarkers and therapeutic targets. There are certain research questions I have identified in this paper. The first question is what are the specific proteomic changes in the tear associated with the different OSDs? Second one is the proteins are differentially expressed in the tears of the patient with follicular surface disease and it is compared to the healthy controls. Third one is how the identified protein correlate with the clinical parameters of the OSD. Fourth one is what are the functional rules to the identified protein in the context of the OSD. Fifth one is how effective is the method which we are using for the biomarker discovery and validation in the tear proteomics. So the objective of the manuscript is first to identify the specific protein that are differentially expressed in the tears of the patient who are suffering from OSD and then compare to a normal patient. Second one is the aim to find the proteins in the tear sample that could serve as a biomarker for the presence in the OSD. Third one is to correlate the specific proteomic changes with the clinical features like the symptom severity or disease stage. Third one is to understand how the differentially expressed protein might contribute to the progress of the disease. Fifth one and the last one is to find the LCMS and with more extensive method like multiplex and that assess the relevance to the patient management. Now let us study what basic tear proteomics is. Basic tear proteomics is a study of protein present in the tear fluid using proteomic technique and it helps in understanding the protein composition of the tears and how it responds in order to the patient management. The tear proteome film analysis is a powerful analytical tool which might be able to establish the underlying pathophysiological process. And this pathophysiological process provides diagnostic to the biomarkers using label-free LCMS method. Then let us learn about largely carbonyl functional unit. This is basically known as LFU. So this LFU is responsible for the tear producing and preventing abnormal evaporation of the tear from the eye. Then disruption of this LFU might result in the loss of tear film integrity and normal function and this is caused due to the dry eye. Disruption of LFU might lead to the tear hyperosmolarity. Sometimes this follicular dryness promotes squamous metaplasis. So what is squamous metaplasis? Squamous metaplasis is to evaluate the degree of the damage of the conjunctival epithelium, which is the impression of etiology to skin. Tear film proteomes identify biomarkers of the aqueous deficient dry eye and the meibomian gland dysfunction. This is the main cause for the evaporative dry eye. There are six methodologies to follow the process. So the first one is sample collection, where we recruit the patient with a specific OSDs like DED and MGD, along with a healthy person. Then we collect consent form from them to prevent further ethical issues. Then the tear is collected using standardized methods for tear collection such as micropipettes, and it is stored in minus 80 degrees Celsius for integrity and until analysis. Then the sample is prepared for the protein extraction using the required buffer. Then it is quantified the total protein concentration using assays. Then the proteins are digested into peptides using enzymes like trypsin and automated digestion system. Then the third step is for label-free LCMS analysis. In the uh, LC analysis, first we separate peptides using high-performance liquid chromatography, which employs reversed phase column for peptide separation. And the second step is the mass spectronomy, where we perform tandem mass spectronomy, which is to identify and quantify the peptides. Then the data is collected in the label-free quantitative mode, focusing on the peptide abundance for the quantification. At last, we analyze the MS data using softwares like Mascot. So, what is a Mascot software? Mascot software is a software Mascot is a software or a search engine that uses mass spectronomy data to identify protein from the peptide sequences to a database. The fourth step is to biomarker validation using multiplex microarray, where first microarray is designed, then sample is prepared, then direction and quantification is taken place. At last, using Apex quantitative data is analyzed. So, what is that Apex quantitative? Again, Apex Quantitative is a software that provides researchers with the ability to quantify proteins observed in LCMS methods. Then, fifth one is functional and statistical analysis. So, functional analysis is to investigate the biological relevance of the identified and validated biomarkers, and to it, the appropriate statistical of the findings is known as statistical analysis. Then the reporting and interpretation is taken place. So, integrating the data from LC and microarray to provide a comprehensive view of your proteome, validate the key biomarkers in the study, and then we either prepare and publish it as a paper or prepare and publish the finding with the research community. So according to the research paper what I have got, there is one case study where out of 70 patients, they divide the patients into the two parts. One is the discovery phase and the second one is the validation phase. In the discovery phase, 7 people are suffering from dry eye, 12 people were with chronic MGD, and 18 people were normal volunteers. In the validation, 24 people are suffering from dry eye and 9 people are suffering from volunteer. 9 people are normal volunteer. Here, 3 candidate biomarkers are validated through orthogonal technique. Now, what is orthogonal technique? Orthogonal technique is a technique of using independent DE and CT tier samples. So what is the step? First, we identify the protein, then discrimination between dry eye and MGD, then we use LCMS method, and then counting of this thing happens. There are many new techniques and instrumentations coming in this advancements. So first, I would like to go to the advancement instruments. Instruments. So advances in mass spectrometry. The first one is orbit trap fusion luminance that offers enhanced sensitivity and the resolution, which improves the detection of low abundance protein and peptides in the tear sample. Then TIMSTED. TIMSTED provides mass accuracy and resolution by incorporating trapped ion mobility spectrometry with time of flight mass spectrometry. Then we have data independent acquisition, that is DIA method with SWAT MS. SWAT is sequential window acquisition of all theoretical fragment ion mass spectra, which enables comprehensive proteome analysis by capturing the fragments ion information for all precursor ion with defined windows. with the help of enhanced liquid. Next is enhanced liquid chromatography, which is nano LC with column selective switching. Nano LC with column selective is an advanced nanoliquid chromatography with column switching technology, which improve the peptide separation efficiency and sensitivity. In the techniques, it is microfluidic devices, which is on-chip sample preparation. The on-chip sample preparation is a device which enables automated high-throughput sample preparation, which reduces the sample loss and contamination. In the ultrafiltration, it is a method which helps in concentration of low abundance protein and remove contamination before LCMS analysis. Then prefactoration is a method like isoelectric focusing and size exclusion chromatography. Machine learning and AI is improvement of new AI tools. Integrative bioinformatic tools is proteomic data integrative platform, which is the tools like Max Quanta, Proteome Discover, and the Skyline, which are integrating proteomic data and additional omic layers. Network analyzing tools like Cytoscope and Stream, which is again for omic layers. Now, as we are coming to the result is being split into the research questions. So first is biomarker candidate selection, where patient with dry eye and MG were analyzed with a symptom as OSDI, and the tear volume assessment to ascertain the subtype classification using Scheimer test and to damage and the damage to the ocular surface as standard test. Here, the squamous metaplasis grade is given in the table given below. The second one is validation process to evaluate the correlation between biomarkers and clinical parameters like OSDI, TBUT, schema 1. So TBU and the fluorescence staining were obtained for each patient. TBUT is a tear breakup time, and all the parameters were different significantly between the DE patients and CT individuals. And for the timing is given in the second page for the validation process. Then if we are moving on to the third one, that is the protein analysis. In the protein analysis, we have analyzed 26 kinds of protein, starting from complement C3 protein S100A6, protein S100A8, ceruloplasmin, apolioprotein, alpha-1 acid glycoprotein 2, and much more. Here, we find the highest mean, lowest mean, and number of peptide in certain proteins. So this is the table for the quantitative analysis using spectral countings. In the conclusion, we are able to conclude the integration of the label-free LCMS and multiplex microarray technology provide a comprehensive approach to the understanding of the tear proteome. With the further research and validation are needed to be established for these biomarkers and the clinical practices. So the key findings in this manuscript is to identify the dry eye disease with elevated inflammatory marks and altered protein composition, MGD along with conservatives. Also find the insight into the disease mechanism in inflammatory pathway along with activation of inflammatory pathway, disruption of tear film components, and etc. What I have learned from this manuscript is a protein identification and quantification using LCMS and label-free method. Then disease biomarker, that is comparing of proteins of a tear from the healthy individual from those of a patient with OSD, and you can identify the specific protein that are differentially expressed. Then we have pathophysiological insight, then validation of biomarkers, disease and classification with severity and the progress, and in personalized medicine. That is to understand individual variation in tear proteome that contribute to the personalized medicine, where treatments and intervention are customized based on the specific proteomic profile for for a certain patients. And then, what are the future research plans if I have to do? That is longitudinal studies, that is to drive the changes of protein over time with patients with ocular surface disease to understand disease progression. Then is to expand sample size and diversity. Then is to do a comparative proteomic study where your proteome across different OSD and other related conditions are compared. And then the fourth one is to integration of other omics data, like proteomic data with genomic, transcriptomics, and metabolomics to gain a more holistic view. Then validation and functional studies, where validated identified biomarkers in larger clinical trials and investigate their functional roles in disease pathology. Then development of diagnostic tools, and then at last personalized treatment approach.